Drum roll, please. All right, waiting for the official beep, the official ding. Sorry, I'm seeing a little. There we go. There's the official ding. Okay, so we were talking about platyhelminths. And the subgroup within the platyhelminths, the Neodermata. And as I mentioned earlier, these develop a secondary epidermis that doesn't have cilia on it. And that epidermis is a syncytium. And you've seen that term before, and I told you we would need it later on. A syncytium is what you get when either you have a cell's nucleus divide but not the cytoplasm, or when separated cells come together right now over me, like John Lennon said, and fuse their cytoplasm but maintain separate nuclei. Either way, what you get is basically a single giant cell with multiple nuclei. That's a syncytium. Syn is together, cyto means cell. Syncytium is a together cell. And when we come to it, I'll show you why that's important, because it turns out to be very useful for parasitic lifestyles. All of the neodermatans are parasitic, although not all of the flatworm parasites are neodermatans. There are some parasitic flatworms that are not in the neodermata. This is one of them just because it looks cool. These are either parasites or commensals, mostly of crustaceans. They're the kind of things you could conceivably find on crawfish if you scraped at them enough. They're flatworms with a sucker on the posterior end and these little tentacles sticking out of the head. And yeah, they look cool. They look like little squid, but they aren't. They're very similar to planaria worms, except they happen to have tentacles on the head. So that's just an example of a parasitic flatworm that isn't a neodermate. In fact, this is some pretty recent cladistic analyses based on the entire genome, which you can do these days. It's become so relatively cheap and relatively simple to sequence DNA that you can sequence entire genomes now pretty easily. And down at the bottom there in the red box, those are the three taxa that belong to the Neodermata. The Cestoda is tapeworms. Monogenea is that ectoparasite group that hangs out on fish. And Digenea is flukes. And they all form a clade. Above that, you have other groups of platyhelminths, including a number I didn't even talk about. You know, I mentioned I was not going to require you to know most of those. But the three that are outlined in green have at least some parasitic members. So there's at least three groups of free-living flatworm that have some members that have turned to a life of parasitism. The one in the middle, the Ficampiida, can actually be a rather serious pest of commercially important clams. Don't know how many of you want a career, marine clams. Don't know how many of you want a career as a clam digger, but in case anyone does, ficampids might be the sort of thing you might have to worry about. So parasitisms evolved at least 4 times in platyhelminth evolution, but the Neodermata are by far the most successful at it. And we can divide these into monogenids, trematodes, which includes flukes and a funky little group called Aspidobothrians, which means shield with a ditch dug in it, and I'll show you why. And then Cestoidea is the true tapeworms, plus some oddball things that nobody understands that tend to live in the rectums of chimera fish called cestodarians. And we'll talk about those in a little bit. And that's what that secondary epidermis looks like. This happens to be a tapeworm, but the basic anatomy would be pretty similar in monogeneans and flukes as well. This thing will start life with a ciliated epidermis, and then these cells that are underneath the epidermis and under the muscle layers, the big blobby thing that has the nucleus and the nucleolus labeled, those will actually send up little extensions, and the extensions will go through the muscle layers and cover the old epidermis and spread out and fuse with each other. So what you have is an epidermis where these extensions of the epidermal cells form this covering over the entire body, and the actual bodies of the epidermal cells are down below, under the muscle layer. That cell body, with the nucleus it's called a cyton, if you're curious, which is cool because I always thought cytons were the ones that were trying to kill the humans in Battlestar Galactica, but I guess I was wrong. And the cytons send out these extensions up through the muscles, and they spread out over the cilia, and they cover the old epidermis, and they fuse to form this continuous sheet of cytoplasm. That can be useful for things like nutrient absorption. This happens to be a tapeworm, and tapeworms, although not others, that secondary epidermis is covered with little hair-like structures called microtrichs, Greek for small bristles. That increases surface area because tapeworms just absorb food directly from the gut. They don't have guts of their own. They don't eat. They probably help the worm adhere to the body wall. Another reason that arrangement's useful for parasitism is, imagine that I could get my fondest wish and I could get to operate a big wrecking ball and knock Lewis Science Center down so that it could be replaced with something that's actually livable. I'm sure you've all thought of it too. And you have a wrecking ball hitting a brick wall. Where it's going to crack is probably going to be where the brick meets the mortar, right? Much of the cracking is going to be on the boundaries between adjacent bricks, or the boundaries where one type of material, the brick, touches another type of material, that's the mortar. That's a problem if you're made up of multiple cells. It's possible for enzymes and possibly even, antibodies or white cells and things like that to work their way into the cracks between your cells. If you're a parasite, you've got to come up with a way to keep from getting digested by your host or attacked by your host's immune system. And if you've got junctions between the cells on your outside, that's a weakness that they could exploit. You could have, your host white blood cells squeezing between your cells to wreak havoc. White cells can squeeze between cells in your body. It happens all the time. Or you might have enzymes that can get into the crack between cells and attack maybe cell surface proteins or something like that. The problem goes away if you don't have any boundaries between adjacent cells. If your entire outer covering is one single cell, there's no cracks that your host defenses can jimmy open. And so you'll see this a second time in another big time group of parasites, the Acanthocephala, which may have to wait until they'll have to wait till Monday. But that's the function of that secondary epidermis, which, by the way, is also called the tegument of a tapeworm, fluke, or monogenean. It is pretty adaptive for a parasitic lifestyle. The trematoda are the so-called flukes. Well, they include the flukes. Trematodes include digeneans, the true flukes, and this odd group called the aspidobothrians, which I'll show you in a bit. They're typically endoparasites, either inside tissues or in blood. There are some flukes that inhabit the bloodstream. There's others that burrow into liver, lungs, bile duct, places like that. The adult's got a pair of suckers. One surrounds the mouth and one's down on the belly. That's the oral sucker and the ventral sucker. These allow attachment to the tissues. The oral sucker may also allow the fluke to slurp up mucus or blood or tissue or whatever it does. And the life cycle involves multiple hosts. This can vary immensely, but typically the intermediate host, the one where sexual reproduction doesn't take place, is a mollusk. Very commonly, that's a snail. Eggs will pass out of the definitive host body, and typically they'll get washed into water or they'll fall into some kind of moist habitat, and the larvae will burrow into a snail. Or occasionally it's a clam, but snails seem to be the most common. And they'll go through several stages within the snail. So the snail is what we call the intermediate host. They'll reproduce asexually in the snail. And then when the time is right, they'll burst out and penetrate into usually a vertebrate. And in the body of the vertebrate is where they'll undergo sexual reproduction. So if sex is happening in a host, we call that host a definitive host. And then asexual reproduction takes place in an intermediate host. And again, I mentioned this earlier, but it bears repeating. The good thing about sex is that it generates biological diversity. The bad thing about sex is that it takes time. You've got to find a partner. You can't just start budding off offspring as fast as you can. You have to find a partner. There's mating to be done. Sexual reproduction is generally slower. You can't hurry love. No, you just have to wait. Love don't come easy because it's a game of give and take. And that's not a problem if you can reproduce asexually. So just to mix up my pop culture metaphors, a lot of organisms alternate sexual and asexual stages so they get the best of both worlds. They can chill it out, take it slow, then they rock out the show. Too early in the morning for me to try to sing that. Anyway, so you'll alternate sexual reproduction in a definitive host and asexual reproduction in an intermediate host. You get this in the true flukes, the digenea. You don't get that alternation of generations in the first subgroup of trematodes, the Aspidobothria, which look like this. I won't say very much about them. It's a small group. They've got no known medical or veterinary importance. They don't parasitize anything that people care about. As far as I know, they don't seem to do a great deal of damage to their hosts, and they can actually live outside for long periods of time. So this one has, if you look down at the lower right, there's a thing labeled BF. That's the mouth, surrounded by a funnel-shaped sucker called the buccal funnel. And then the sucker on the belly side is immensely expanded, and it's actually divided up into compartments. Aspidobothria means shield with a trench, and now you can see why. That great big shield on the ventral side, that big sucker, looks like a shield that's got a bunch of trenches dug in it if you're on drugs. And that's all I've got time for with those. True flukes do have a life cycle with asexual and sexual reproductive stages alternating, and virtually always the asexual stages take place in a snail, the so-called intermediate host. You're looking at an adult right there, and an awful lot of the colorful structures that you see are gonads. I believe that's ovaries in, shoot, I always get this confused, but I think it's female structures in very dark blue and testes in orange, I think. Don't quote me on that. But much of this guy is filled with reproductive organs. One that's been studied a great deal is the sheep liver fluke. This one you can see there's a pair of suckers. One of them is at the very tip at the top, and it's actually cut off a little bit, but that surrounds the mouth. And then below it on the ventral side, there is a little perfectly round pink thing. That's the ventral sucker. Most of what you were seeing that worm full of is reproductive structures. An adult fluke will live in the liver or the bile duct of a sheep, sometimes goats, occasionally humans. They can infect us, although they don't very often, and will just slurp up mucus cells and blood. Back of the mouth, there's a sucking pharynx that can suck strongly enough to damage host tissues. And that's what the anatomy looks like. In brown, that's the gut. You can see the oral sucker surrounding the mouth, and then the gut leads into a pharynx, which branches. You have two main branches of the gut right there. In dark blue, you've got the female structures. The branching thing over on the left is the ovary. The ovary produces eggs and releases them into the oviduct. Within the oviduct, the eggs get plastered by yolk-containing cells that are produced in these very branchy things at the left and right called the vitellaria. So the egg gets a supply of yolk, it gets a shell put on it, and passes through that long oviduct to the outside. In light blue, you've got testes and a pair of structures called vasa deferentia. You know, that's the plural of vas deferens, you know, which some of you also have. Those convey sperm to the male genital aperture, and the openings of both of these are located at the tip of a retractable thing called the genital papilla. Typically, these worms will not self-fertilize if there's another option, but they'll put papilla to papilla, and each one will fertilize the other's eggs. So yes, some people think there's not, that male fertility and male sterility are the same thing, but the truth is there's a vast difference between the two. Right, sorry. Okay. Digynean life cycles can get pretty complicated, but a simple one would go like this. Eggs will pass out of the definitive vertebrate host, often in the feces. If you've got a fluke living in the liver or the gallbladder or the bile duct, the eggs often pass out in the feces. The eggs will hatch into larvae, and larvae infect the first intermediate host, which is almost always a snail. In some cases, the snails eat the eggs, and the eggs hatch inside. In other cases, the eggs hatch into little ciliated larvae called myricidia, which can swim for a while until they penetrate snail tissues. They'll just burrow right inside. Either way, once they're in the snail, the myricidium larva becomes a sac-like structure that hasn't got a mouth. It's called a sporocyst, and it starts budding off offspring inside. So it reproduces asexually, and in the life cycle we're going to look at, the sporocyst produces a whole bunch of a new stage called redii. And you can tell the difference because redia do have mouths and they can feed. The sporocyst depends purely on absorbing food from the snail through its tegument. And so the redia come bursting out of the sporocyst, redia not, and each redia can feed. It's got a mouth and a gut, but each redia reproduces asexually again and produces multiple types of a larva called a cercaria. And cercariae you can usually recognize because they almost always have a tail. And in the species we're going to look at, the cercaria will leave the intermediate host. They can come bursting out of a snail and they'll just start swimming. In some species, the cercaria will just burrow right into the definitive host and metamorphose into an adult. In other species, the cercaria will form a resting state called a metacercaria. And the metacercaria will just sit there inside a resistant wall, forming what's called a cyst. In the species I'm going to talk about next, the metacercaria forms on, in the sheep liver fluke, the metacercaria forms on a blade of grass or other plant, and then the sheep is infected when it eats the grass. In one that I've seen in my parasitology class, the metacercaria will, or the cercaria will leave a snail and burrow into the tissues of a fish and form resting cysts. They'll form metacercariae sometimes on the outside of a fish and sometimes inside the fish's liver or something like that. And they'll just sit there and wait to be eaten by, in this case, a fish-eating bird, which is the definitive host. You can actually look for this. I don't know how many of you enjoy going fishing, but there is a pretty common parasitic flatworm that grows and matures in snails. Cercaria burst out of the snails, and they'll burrow under the skin of a fish and form a resting metacercaria. And the thing that the fish does is part of its immune response is to secrete melanin around the metacercaria, which means that the fish looks like it's covered with black spots. And in fact, this is called black grub or black spot disease. And as far as I know, it's not common in freshwater aquariums, but it's the kind of thing that you might see in the fish that you pull out of Lake Conway someday, if any of you folks go fishing. There's other cercarias that can form kind of yellow lumps on the fins. That's called yellow grub. There are cercaria that can form metacercaria on farmed catfish that make these unsightly red swellings in the muscle that make the fish unsellable. That's called red grub. That's the kind of thing that catfish farmers in the Mississippi River Delta have to worry about over in East Arkansas. And there are some that will form metacercaria inside the fish liver, and all of these will just wait for the fish to get eaten by the definitive host, a fish-eating bird. Right. There's lots of possible variations on that theme. Not all species have metacercaria. In some species, the sporocyst might give rise to daughter sporocysts. In some, the sporocyst might just give rise to cercaria and skip the whole redia business. There are other variations possible. In some, there is no metacercaria. It depends. It can get pretty diverse. So there's a diagram of what this looks like in the case of the sheep liver fluke, Fasciola hepatica. That's an adult on the left. The adults will reproduce sexually and lay eggs. And the eggs give rise to, they hatch into these little ciliated miracidium larvae. And the miracidium penetrates a snail, burrows into its tissues. They've actually got little enzyme glands up there at the front, so they can do that. And the miracidium will transform into a sporocyst, which is basically just a big bag that has no mouth. The sporocyst will internally start budding off redia. You can tell the redia because it does have a mouth and a simple gut. And inside the redia will be produced cercaria. This is a stage that has pretty much all of the adult features, and it's got a tail, and the cercaria is capable of swimming. In flatworms that spend all of their time in water, the cercaria may just swim until it infects a definitive host. But in the case of sheep liver fluke, when cercaria come bursting out of the snail, they curl up on blades of grass and form metacercariae, a resting stage, which can last for, I think, several days before they get eaten by a sheep or a goat and transform in the sheep or the goat's body into the adults that will chew on the sheep or goat's liver. Incidentally, humans can get infected by sheep liver flukes. It does happen. It's fairly rare because humans don't typically eat grass. But you can get metacercaria on other things that humans eat. It's not really common here, but in Britain, people eat watercress. That is an herb that grows in streams and has a rather nice peppery flavor. It's commercially farmed in some areas. The Brits do like their egg and cress sandwiches, egg salad with watercress on top. And occasionally, if your watercress is growing just a little bit too close to a sheep pasture, you'll get metacercaria forming on the watercress. They can form metacercaria on pretty much any plant, and people that eat contaminated watercress occasionally will get sheep liver flukes, although it's not especially common. So that is very common type of life cycle, although, as I mentioned, sometimes you might skip the redia. There might be more than one round of sporocyst formation. There may not be metacercaria at all. Sometimes the snail eats the eggs directly and the miracidium isn't seen, at least not free living. A lot depends on what species you're talking about. And there's some that do really weird and frightening things. These are redia of a fluke in the genus Hemathsla. I forget what the definitive host is. It might be a fish, but the intermediate host is a mud snail on California mudflats. And if you crack open some snails and dissect them out, if they're infected, you can find two different types of redia. Over there on the left, that's a primary redia, which is this huge sac-like thing, and I can actually see at least 5 cercaria inside that retia. The retia is basically a bag of cercaria. And yeah, I can see that each one has, two suckers and a tail. They're kind of tadpole shaped, but all smushed together. The secondary retia doesn't produce cercaria and will eventually die without producing any offspring at all. Over there on the right, that pH is where the pharynx is. So that's where the thing's mouth is. These do actively feed, and what they feed on is unrelated retia. So if you have a snail that is infected by, two different parasites at the same time, each of those will develop into sporocysts. Each sporocyst develops multiple retia, and the retia from one will attack and kill and eat retia of the other. So you have reproductive retia and hunter-killer retia that clear space to make sure that only their retia get to reproduce. They'll kill unrelated retia. So this is really cool. Two families of parasites desperately fighting for dominance inside a snail. Like, dang, talk about, feeling a war inside yourself. So yeah, this is really weird. It's a fluke that has social castes, right? Just like ants have workers and warrior castes, and termites do. Here you've got Digenean flukes that do. Very strange things these guys get up to. One of you was telling me that you've seen this before. That's the European amber snail on the left. And that's a normal one. It has small tentacles and it can retract all of its body into that shell. Snails have been known to graze on bird droppings. And if they do that, there's a risk that they will eat the eggs of Leucochloridium paradoxicum. Once inside the snail, the eggs hatch into sporocysts that enter into the tentacles and develop into these giant pulsing things with rippling color bands. So the snail on the right, both of its tentacles are grossly swollen, filled up with these giant pulsing sporocyst sacs. And the snail can no longer pull those in. Now, since the snail is, can't retract and has these pulsing sporocyst sacs that look just like a tasty grub or a maggot, the snail is quite likely to get eaten by a bird. And then Leucochloridium paradoxicum promptly proceeds to parasitize the bird. And the bird will pass out eggs in its own droppings. The droppings fall on a leaf, get eaten by a snail, and it's the circle of life. And it moves us all. How does that go? I don't remember. Anyway, right. And yeah, I need to remember to post some footage of that. There's even flukes that manipulate host behavior. This is the lancet liver fluke. This is another one that is capable of parasitizing people. They'll go through the usual stages of life cycle in snails. When the cercaria come out, they'll penetrate into ants, and a metacercaria of dicrocelium forms a cyst on the ant's nerve center, on the ant's brain, we'll call it that. And the ant starts acting weird. When the temperature is low, an infected ant will start crawling up to the tip of a grass blade instead of staying down below like normal ants would. And it'll crawl up there, lock its jaws onto the tip of the grass blade, and that increases the likelihood that it will be eaten by a grazing sheep or goat, and then the sheep or the goat gets parasitized by dicrocelium. So I'm not quite sure how it does the trick, but those metacercaria are able to change the behavior of the ant and make the ant act in a way that does not favor its own survival, but favors the survival of the host. Now, parasites can make hosts do weird things sometimes. That's not the only case of this that we'll see. Human trematodes tend not to be common here, not in the West. You tend to get them in tropical countries where sanitation can be a problem, and or when eating certain foods without full preparation is practiced. I mentioned that the sheep liver fluke can sometimes infect humans if they eat contaminated watercress or I guess some other vegetables. I guess you could get it from your spinach if you had sheep close to your spinach patch or something like that. Asian liver flukes can be transmitted by eating fish that is not cooked or not fully cooked. Lung flukes you can get from eating freshwater crabs or crawfish. In some cultures, freshwater crabs are eaten with minimal or no cooking, and you can get lung flukes that way. Schistosoma is transmitted by contact with water with cercaria in it, and I'll talk about that one in a bit. This is the kind of thing you could conceivably experience if you go swimming in a place like a pond or a lake that's got lots of snails in it. When the temperature is warm, the snails will start releasing cercaria larvae, and sometimes they'll try to burrow into human skin. This usually doesn't cause problems for humans. They can't burrow deeply. They can't infect you by burrowing through the skin. What they can do is cause a kind of contact dermatitis, affectionately known as swimmer's itch. So if you have ever gone swimming in the old swimming hole or Lake Conway or Lake Beaver Fork or anything like that, if you've ever gotten out of the water and ended up with a bit of a rash, that might have been caused by cercaria of aquatic flukes trying to burrow into you. Fortunately, they can't succeed. They're probably adapted to burrow into fish or possibly duck feet or something like that. So yeah, if they get you, it's a dead end. But this is something you could conceivably experience if you spend a lot of time in, grandma's old swimming hole or something like that. And one of the worst problems for public health in certain countries is flukes in the genus Schistosoma. Schistosoma mansoni right there is one of 5 species of human blood fluke. And unlike many trematodes, the sexes are separate. And I actually messed up the caption of this. The female is this very elongated form that lives in a long, narrow groove on the male's body. That's an error that I need to correct. So up at the up towards the top, that's the head and the ventral sucker of a male. The male's got a long groove on his body called, I think, the gynecophoric groove, and there's a female threaded right into his gynecophoric groove. She has followed the advice of Madonna in 1984 and gotten into the groove. And you kids don't remember that song. Damn millennials, avocado toast. Right. So they'll live paired up like that, and humans are the definitive host. And you'll find them in, depending on the species, they might be in the blood vessels of the liver, the blood vessels of the intestines and spleen, or sometimes in the blood vessels surrounding the bladder. From there, eggs will pass out either in urine or feces. The eggs might kind of have to work their way through some tissue, but they'll pass out in urine or feces, fall into water, And the eggs will hatch in water into miracidium larvae, which will penetrate into snails. The snails will go through the usual stages. Each snail will, thousands of cercaria emerge, and then the cercaria themselves directly penetrate the skin. I mentioned that people who get swimmer's itch are being attacked by cercaria that can't get through the skin. This would be true for most of the cercaria that you would encounter here. Schistosoma cercaria, however, can penetrate the skin. And if you are in places like, I think they advise tourists not to contact water of the Nile River in Egypt, because that's a good way to get schistosoma parasites, is to wade or swim in the Nile. You end up with cercaria penetrating in, and they can actually permanently infect you. So there's an egg of Schistosoma mansoni on the left. The egg shape, those of you going to be doctors, you may never need to know this, but if you do tropical medicine, you may need to tell species apart. Some species don't have a spine, others have it at one end, and Schistosoma mansoni always has that spine coming off at about a 10 o'clock angle. Those eggs will hatch into metacercaria. The eggs will hatch into miracidium larvae. The miracidium will reproduce in a snail. And then the cercaria on the right come bursting out of the snail, and that's what will penetrate the skin of a human that's immersed in the water. That darker staining part up at the front is virtually a miniature adult, but behind it you have the tail. And schistosoma and its kin have forked tails. If you must know, this is called being Furca circus, but don't worry about that. Not critical here. So the cercaria will burrow in, and eventually they'll migrate to the circulatory system. Exactly where depends on the species that you're talking about. That's a cross-section through a blood vessel where you can see a couple of schistosomas, each of them paired up. There's at least, I guess that's just one pair, hanging out in the blood vessels. They lay eggs. Eggs manage to work their way through the tissues, and we don't fully understand how. But ultimately, they pass out of the human body through urine or through feces. What this looks like depends on what species of fluke you've got. If you've got Schistosoma hematobium, they love to live in the blood vessels surrounding your bladder. So they'll live there, they'll lay eggs. Eggs will work their way into the bladder, causing scarring and tissue damage as they do, and ultimately pass out via urine. Brace yourself for the next slide, please. And it looks like this. A very common symptom of Schistosoma hematobium infection is bloody urine. There are, I believe it's a group in Africa where the men spend lots of time wading in irrigation canals. They farm using irrigation systems, and those canals have to be maintained. So if you're a man in this particular group, you spend a lot of time wading in water. Well, guess what else likes to live in irrigation canals? Snails, which means that men are typically chronically infected with Schistosoma hematobium, and boys begin working alongside their fathers at about the age of 12, which means that every boy in the tribe starts getting bloody urine at about the age of 12, and this is considered the equivalent of menstruation. Women begin bleeding from their private parts at about the age of 12. Why shouldn't men? It's just kind of normally accepted as part of the life cycle. I mentioned that this is a problem in Egypt. The ancient Egyptians actually did have this, and it's documented in one of their old medical papyri. So this has been a problem for humans since at least 2000 BC or something like that. Not only is the bloody urine no good, but scarring in the urinary tract from these eggs working their way through the bladder wall can ultimately block the urinary tract. It can lead to secondary infection by bacteria, and it increases your risk of bladder cancer. So there, pretty good reason to try not to get schistosoma hematobium. A different species is Schistosoma mansoni that prefers the blood vessels around the intestine. Eggs there pass out in feces, and they tend to spread in areas where the sanitation's not great. If you have human waste entering a water supply that's got snails in it, the parasite can complete its life cycle. And at its worst, this can lead to something called hepatosplenic schistosomiasis, where blockage of small blood vessels causes gross swelling of the liver and spleen. That's liver, not liber. That's a typo on my part. And here you've got a couple of children with badly swollen livers, causing the entire abdomen to swell up as a consequence of hepatosplenic schistosomiasis. Another good reason to avoid this. Very simple way of breaking the life cycle is simply watch where you pee and you poop. You can break the life cycle if you just don't allow eggs passed from an infected human to ever get anywhere close to a snail that they could infect. And I told my parasitology class this: if you want to grow up to save lives, don't be a doctor. Design low-cost, easy-to-install human waste treatment systems for the third world, and you'll probably end up saving a lot more people in the long run, because a lot of these are transmitted in human populations that aren't able to treat human waste efficiently. There's other things that can contribute to it too. But yeah, quite possibly the best way you could save a lot of lives cheaply is just to come up with efficient and simple ways to break parasite life cycles. And one way of doing it is just, come up with a way to make sure nobody pees in the same pool that they're swimming in. So there. Right, that was a little unpleasant. Monogeneans, or monogenoidians, are... Actually, it's 10:52. How about I give you guys another break? This is a good time to take a break. So I will go ahead and sometimes when I stop, I can't find the icons I'm supposed to click on because I'm working in this dual screen environment that's a little confusing at times. About the recording, stop now.